Biblical DNA here, and we have a new paper regarding the genetic history of Scythia. If you've watched my previous videos on who the Scythians were in the Bible, they were known as the Ashkenaz. That's the Semitic word that was used to identify them by the Akkadians, the Assyrians, and the biblical writers. All right, so you can find that in my description of my previous videos if you haven't watched them. They're also mentioned in the New Testament or Brit Hadashah in Colossians chapter 3 verse 11. They were considered an uncivilized people. However, let's get into the actual archaeology, anthropology, population genetics of who the Scythians were. So, genetic history of Scythia, Andreeva et al. 2025. It literally just was published, I think, 2 days ago, if I'm not mistaken. So yeah, I saw it as soon as it dropped, but I was like, I don't know if I want to do a live on it because I've already covered them, and it seems like there's so many Scythian papers that are dropping. So I'm like, should I cover it? It's like, should I cover all the Roman papers? So let's get into this. Great Scythia was the ancient Greek name for the area stretching from the northern Black Sea coast to the middle Don, using high-quality genomic data generated from 131 ancient individuals from Great Scythia and neighboring regions of the Bronze Age and the Iron Age. We established the genetic structure of the Scythians, revealing their diverse origin with major European Bronze Age ancestral components and genetic traces of migration and invasions. We uncovered relationships between Scythians, including elite Scythians, substantial endogamy in the Scythian clan was found. We examined Scythians' phenotypes and medical genetic background and found a harmful gene mutation causing fructose intolerance. This ancient Scythian mutation has spread throughout West Eurasia and has become the most prevalent genetic cause of fructose intolerance in contemporary European populations. Introduction. The early Iron Age in the Eurasian steppe was associated with the renowned Scythio-Siberian cultural region spanning from Inner Mongolia in the east to the northern Black Sea region in the west. The nomads of the Scythian world played an important role in shaping the political, economic, and cultural dynamics during the Iron Age. This population was united by similar or shared cultural traits and included both the European classical Scythians described by the ancient Greeks and the Siberian and Western Asian tribes known as Scythio-Siberian nomads. In the 5th century BCE, a Greek historian named Herodotus described in detail the lifestyle, diet, and phenotype of the classical Scythians from Great Scythia. Herodotus Great Scythia stretches from the northern Black Sea coast to the upper reaches of the Dniester River and to the middle Dnieper River and its northeastern periphery compresses the lands of the right bank of the Don River. The numerous burial mounds left by nomads here during the Scythian era, 7th century BCE to the beginning of the 3rd century BCE, provide a unique opportunity for paleogenetic studies. Scythian burials can be recognized by certain archaeological indicators. These include animal style, artistic tradition, and various artifacts, specific styles of or types of weapons, including the composite bows and Arsenacis daggers, and distinctive horse harnesses, e.g., bridles, bits, and saddles. Together, these three distinctive cultural components are referred to the Scythian tribe. Here, we conducted the genetic study of the Scythian tribes that inhabited steppe and forest steppe regions from the lower Danube, North Pontic steppe to the Don River, as well as in the northern region of the Caucasus. The Scythian culture indicates inhumation rites and burials that are predominantly in the form of mounds. These burial sites are primarily connected to the paramilitary segment of Scythian society. There are two main hypotheses regarding the emergence of nomadic Scythians in the North Pontic region in the 7th or 8th centuries BCE. According to the first one, the Scythians formed on the basis of both local, agricultural, and migrant tribes of the Shrubnaya culture of the late Bronze Age. The second concept assumes the appearance of the Scythians in their developed form as migrants from Asia. The Central Asian region is considered to be the most likely ancestral homeland of the Scythians and other Iron Age Eurasian nomads. In the periods preceding the Scythians, the European steppes also served as a contact zone between European and Asian populations, representing the area where different cultures replaced each other over time. In the Middle Bronze Age, 3rd millennium BCE, the catacomb cultural historical community spread from the mouth of the Danube in the west to the Volga in the east, and from the North Caucasus in the south to the Upper Don in the north. In the late Bronze Age, 19th to 13th centuries BCE, the Sarumnaya cultural historical community was represented across the vast steppe territory of Eurasia, represented by several local variants. To date, published ancient genomic data for Middle and Late Bronze Ages are available from the European steppe. Nevertheless, no data on their impact on subsequent Iron Age groups from this region. A substantial gap in our understanding of the Bronze Age to Iron Age transition can be addressed with previously unknown genomic data from Eastern European burial sites. To date, a H-Iris-Plex-S system has been developed to predict eye color or human eye, hair, and skin color. In addition to general genetic connections or diversity, it is of interest to see whether phenotypic variations have changed in groups from the Bronze Age to the Iron Age in the same locations. Real quick. make sure this looks clear. Okay, cool. Most of the genomic data published in the date from Iron Age nomads are derived from Eastern Scythian world tribes, whereas data on the classical Scythians from the Great Scythia are poorly represented. Therefore, the origin and appearance of the Iranian-speaking Scythian tribes in the historical arena of the Caucasian and Caspian steppes around the 7th century BCE, early Scythians, remain poorly understood. The disappearance of the Great Scythia at the beginning of the 3rd century BCE also remains unresolved. The specific phenotypes attributed to Scythians by ancient authors, such as bronze skin color, reddish hair, and meat-eating habits, remain in the realm of mythology. The similarity of the physical trait or status of men and women, along with mentioned low fertility and obesity among the Scythians, may imply the presence of endocrine disorders. It should be remembered that most ancient authors incorporated folklore and fables into their historical narratives to enhance their appeal. However, we believe that the history and some myths of Scythians can be attested through genomic approaches. Results. To elucidate the origin and history of the Scythians, we collected samples in 154 individuals from Bronze Age and Iron Age archaeological material from West Eurasian regions corresponding to Herodotus' Great Scythia, and performed whole genome sequencing. Through a quality check of 289 genomic libraries, we were able to obtain data from 85%, n equals 131 of the individuals, with up to 20 times genomic coverage. These data included 18 ancient individuals for whom only mitochondrial genomes were reconstructed. We obtained whole genome data for individuals from the early classical Scythian mounds and late Scythian burials, n equals 78. Materials obtained from sites dated the 7th century BCE to the 1st century CE. These samples originated from 4 distinct locations across the Scythian area, spanning from north to south, namely the Middle Don, North Pontic Steppe, Crimea, and Pre-Caucasian Steppe. In addition, samples from non-Scythian ground burials of the Similuk settlement in the same geographic area of the Middle Don of the Scythian era were analyzed, n equals 8, 4th to 3rd centuries BCE. We also performed genomic sequencing of non-Scythian individuals from the Paternal Borkaska population northeast of the Scythia area. N equals 11, 3rd century BCE to 2nd century CE, which had not been previously characterized by genetic genomes. Koban culture, N equals 7, 8th to 4th century BCE, and non-Scythian Iron Age individuals from the pre-Caucasian steppe. N equals 9, Alans, Sarmatians, Maotonians, 7th century BCE to the 1st century CE, on the southern border of the Scythian world. Our genomic data fill a spatial north-south sampling gap between previously published Western and Eastern Iron Age nomads and cover all temporal Scythian periods. In addition, we collected and sequenced samples from the same geographic area that predate Scythians to test the hypothesis regarding the local origin of the Scythian tribes. Genomic data were generated for individuals of the Middle Dawn catacomb culture. N equals 6 of the Middle Bronze Age, 3rd millennium BCE, and individuals of the Dawn Forest Steppe, Sarmatian culture. N equals 12 of the Late Bronze Age, 2nd millennium BCE. All right. To study Scythians within the broader temporal context of Eurasian demography, we combine newly sequenced ancient Bronze Age and Iron Age genomes with genomic data from previously published ancient and present day individuals using data from the Allen Ancient DNA Resource Database. Genetic structure of the Middle Dawn populations during the Bronze Age. We represent the study of the pre-Scythian Bronze Age individuals of the Middle Dawn catacomb and Dawn Forest Steppe, Sarmatian cultures. We projected ancient individual data onto the genetic variation of present-day European populations. A principal component analysis (PCA) plot revealed that the Middle Dawn catacomb and the Dawn Forest Steppe Sarmatian culture individuals form two distinct genetic clusters, further termed Middle Dawn, Middle Bronze Age, and Middle Dawn Late Bronze Age, respectively, both of which are genetically homogeneous, as indicated by admixture analysis. We found that relative proportions of the ancestral components of Middle Bronze Age differed from those of other previously published early Bronze Age and Middle Bronze Age populations. However, according to the F3 statistic, the Middle Bronze Age group shared more alleles with the Yamnaya and Afanasevo groups than with the other early Bronze Age and Middle Bronze Age groups. Through a comparative analysis of Middle Dawn Middle Bronze Age with Middle Dawn Late Bronze Age and other Late Bronze Age samples from various geographic regions, we found that Middle Dawn Middle Bronze Age had a greater genetic affinity to Late Bronze Age Sarmatian from different locations than to other Late Bronze Age populations. The Middle Dawn Late Bronze Age did not share substantially more alleles with the local Middle Dawn Middle Bronze Age compared to early Bronze and Middle Bronze Age from other locations. In addition, we observed differences in mitochondrial and Y chromosome haplogroups between the Middle Dawn Middle Bronze Age and Middle Dawn Late Bronze Age groups. Although mitochondrial DNA haplogroups U, U2, and U5 were present in both Middle Dawn Middle Bronze Age and Late Bronze Age groups, there was no direct matrilineal continuity between them. Consistent with these findings, the phenotypic traits differed between these two populations. How about that? Using the H-Iris-Plex-S system, we predicted that all 5 tested Middle Dawn Middle Bronze Age individuals would have dark hair and eyes. In contrast, 6 of the 8 tested Middle Dawn Late Bronze Age individuals were likely light-haired, and at least 3 had blue eyes. Together, the results suggest a genetic shift in the Middle Dawn region during the Bronze Age, in line with reported across regional migrations in the Eurasian steppe during the Bronze Age, and that the Middle Dawn catacomb population was replaced by a genetically different forest steppe Sarmatian population. Genetically diverse Iron Age groups of European Scythia. Our sequence Scythians formed at least 4 distinct clusters on the PCA plot, further named Scythia Major, Scythia South, Scythia Crimea, and S era West, and were positioned close to present-day European individuals between the modern Caucasus, North European, and Western Pontic populations. Another genetic cluster encompasses previously published Scythian era individuals from Hungary that are distinct from Scythian groups. This group adopted the Scythian material culture but lacked traditional Scythian burial practices. Go ahead and read the. All right, so figure 1, Scythia map and archaeological sites. A, the Scythia geographic area is defined by the known archaeological site locations, a pink tent, and a white border delineate the region associated with the Scythians, historically referred to as Scythia or Great Scythia during the period of its greatest expansion. B, timeline and temporal distribution of the sequence Bronze Age and Iron Age. It samples grouped by geographical region and archaeological site. The Scythians' territory includes archaeological sites from the North Pontic Steppe, shaded in green, Middle Don, shaded in blue, North Caucasus Steppe, shaded in mustard, and Crimea, shaded in violet. So there we go. Payanborskaya cultural archaeological sites were found in the Volga Kama peach shaded region, which is located in or to the northeast of Scythia. The archaeological material represented in Supplementary Note 1, collected from these places, was used for genetic analysis. The color points correspond to genetic clusters denoted on figure 2. The samples with only mtDNA sequences analyzed are indicated by gray points. So, all right, let's continue reading. And it goes to chronological groups, separating the populations of who they were, locations. Asterisk, this group does not represent the earliest Scythians, 8th to 7th centuries BC, but rather represents Scythians after their renowned campaigns in Western Asia and Egypt. I mean, it's so interesting that they say that because from my understanding, they got turned around in Egypt. I don't know. Like it's so interesting that they wrote that. If let me know what y'all think about that in the comment section, because that's interesting that they would mention them going to Egypt, because from my understanding, they made it that far, but this paper is written very interestingly, like towards like Russia, Europe, really Western Eurasia, but it's not really, but not really, because it's like missing out on their expansion. Like it mentions that, right? It mentions Western Asia and Egypt. Well, why not mention what the Assyrians called them? Why not mention what the Akkadian, like their documents, the primary sources that the Semites, the Semitic speaking populations called these people. So another thing is the city during the Greeks called Scythiopolis. In Hebrew, it's in the description box. It's called Beth Shean or Beth Shean. Beth Shean was a Scythian city after the Israelites were taken out of the land by the Assyrians. I'm not really sure, and if you watch my first videos on this, which are in the description box, I go over all of that. But it's just, to me, it's really interesting that they would mention this part, Western Asia and Egypt and their renowned campaigns, but not really mention exactly what they were referred to by these people in Western Asia. Because the Western Asians were Semitic speakers. So it's interesting that they missed that part in this this peer-reviewed research. I don't know. It's, to me, that kind of bothers me. And I looked into the supplementary material, and they didn't mention it either. They mentioned the Assyrians and whatnot, but they didn't mention what the Assyrians referred them to. So once again, you have to watch my first 3 videos on this topic to get an understanding of what they were called and why they were called that and whatnot. But let's continue. The Iron Age group from the Moldova territory, Glino, comprises individuals who share a genetic background with our Crimea cluster and individuals genetically distant from Scythians. The Glino site was described as a Scythian burial ground influenced by Western material cultures. Together, these results emphasize the considerable genetic variability among European Scythia populations and separate them from previously studied Iron Age nomadic groups of the eastern part of the Scythio-Siberian world, such as Saka, Tigris, Pazyaraks, and early Iron Age individuals from Ali Bel, which are drawn toward Asian populations on the PCA plot. Notably, non-Scythian settled Iron Age individuals of Payana Porskaya and Maternal cultures who were sequenced were also distinct from the Scythians on the PCA plot. Although the Similac individuals are located on the PCA plot within the Great Scythia area, partially overlap with the Scythia major cluster. Overall, these settled populations are situated around the Scythian groups. Admixture analysis revealed distinct genetic profiles among individuals within each Scythian and non-Scythian cluster of our Iron Age cohort. In contrast to nomadic individuals of Scyth Major, Scyth Crimea, Scyth South, and Stavropol Iron Age groups, as well as Karbonskaya population, we do not detect the ancient component related to Neolithic Iranian farmers among the S-era West Pinobarskaya, Similac, and Mayaton individuals. Most of the tested individuals represented a minor Eastern genetic component derived from 2 ancient Siberian and or Eastern Asian ancestries. These ancestral components are maximized in the present-day Nganasan and Han Chinese populations, respectively. Only individuals from the Payan Orbaraska population exhibited a pronounced ancient Siberian genetic component. These Eastern ancestral components emerged in the region of the Great Scythia in the Iron Age and were absent in European steppe populations during the Bronze Age. Admixture analysis revealed that Semilac and S-era West carried small amounts of Western hunter-gatherers component, similar to some European and Baltic region Iron Age populations, which was absent in more Eastern groups. The proportion of this component decreased as the component of Neolithic Iranian farmers increased among the individuals. Okay, so that would say, hey, these people did go through Iran or had a population that they mixed with that was related to Neolithic Iranian farmers. Among the Scythian and non-Scythian groups, the outgroup F3 statistics were consistent with the observed PCA and admixture patterns and revealed a substantial number of alleles shared by the Bronze Age and Iron Age European populations, but not the Asian populations. We used QP Atom to model the potential Bronze Age ancestral components to obtain a more comprehensive understanding of admixture sources in the Scythian tribes and non-Scythian populations. Through analysis, we derived multiple valid 2- or 3-way models showing that up to 90 to 95% of the ancestry of Iron Age groups were attributable to admixtures drawn from different step Bronze Age populations. The best fitted models for most of the Crema S era West Similac individuals included Baltic region Bronze Age and ancestry, in contrast to Scyth major 18 of 22 unrelated individuals were modeled predominantly with step-related Bronze Age sources. The remaining ancestors, up to 30%, were derived from Bronze Age populations with a high proportion of ancient components originating from Neolithic, Iranian, and European Anatolian farmer populations. There were also small contributions from ancient Siberian and Eastern Asian ancestries. On average, these ancient Eastern components comprise no more than 10% of the ancestry of Iron Age groups. However, the Palyan-Borovskaya individuals presented a more pronounced ancient Siberian ancestry ranging from 30 to 40%. QP Atom modeling revealed distinct admixture profiles for individuals of Scyth South group lacking notable Bronze Age step related ancestry, but all Scyth South individuals contain more than half Southern ancestry sources, including Turkey, Alaha, Middle Late Bronze Age, it's Italy, Sardinia, Early Bronze Age, and even Ethiopia, Malta. Using unified Bronze Age sources, most of the Iron Age individuals can be modeled as a 2- or 3-way admixture of step, Late Bronze Age, Dawn, Middle Bronze, Late Bronze Age, okay, and ancient Siberian, Eastern Asian, Russia, Shamanak, early Bronze Age sources, with the addition of a Neolithic, Iranian related Uzbekistan, Bastan Bronze Age source. Our findings highlight several aspects of genetic heterogeneity among the European Scythians and their neighboring populations related to geographical, ancestral, and temporal differences. First, no clear geographic genetic specificity was found among European Scythians. For example, the Scyth major cluster combines Scythians from 3 distinct regions: the North Pontic Steppe, the Middle Don region, and Crimea. Similarly, geographical distinct Scythians from the North Pontic and pre-Caucasian steppes are grouped within the Scyth South cluster. The Meotian individuals who were geographically proximate to the Scythians exhibited a genetic profile distinct from the latter. aligning more closely with ancient populations of the Caucasus region rather than with steppe-related groups. In the context of the ancestry of genetically different Scythian and non-Scythian Iron Age groups related to clusters on the PCA plot, we assessed their admixture according to allele frequencies correlation patterns via a cladality test using F4 statistics and outgroup. F3 analysis. We tested a set of populations that may have represented ancestries that present in Eurasia since the yeah. I hate these run-on sentences, and then I want to cover all of this stuff. Early Bronze Age and Middle Bronze Age, i.e. earlier than the Scythians. Okay, figure S7 and tables. Okay, F4 genetic affinity and admixture modeling for Iron Age individuals and groups. A best inferred proximal QP atom models for the Iron Age groups. Each bar represents admixture proportions of the indicated Bronze Age. See details in supplementary note A. Okay. Model rejected at P's greater than 0.05 is indicated by the dark outline for individual models. Okay, turning plot of for 3 or 2-way proximal admixture models for Iron Age individuals using unified Bronze Age sources, Middle, Dawn, Late Bronze Age as a source of steppe-related ancestry, Russia, Shamanaka, Bronze or Early Bronze Age as a source of ancient Assyrian or Asian Siberian ancestry in Uzbekistan, Bastan, Bronze Age as a source of Neolithic, Iranian, Caucasian ancestry. All right. So we see Scythians, B, non-Scythians, C, Iron Age individuals. Okay. Yeah. I don't like when they do this because they should have just done one, explain what it was, and not cut off so much. Okay, so let's zoom in. So they zoomed it in for us from the Bronze Age to the Iron Age of the populations of the Scythians and who they cluster with. Oh, so Ashkenazi Jews did make the cut. They made the cut in the Bronze Age and the Iron Age. What are the odds? Look at there. They're the only Jews that made the cut too. What are the odds? What? I mean, they do cluster closer to Europeans than other Middle Eastern populations, so that makes sense. And it looks like the Scythians cluster towards the Baltics, then Russia and whatnot. Baltic, Moldova, so Eastern European, Russia. It's an interesting base populations components they have for Natufian that I saw. Very similar. Mbuti. So they have a small African component amongst them. A small, I think the Mboti is a Nilotic component in Africa too. I'm pretty sure. somebody can correct me in the video, but I think the Mboti is a pygmy people actually. Yeah, that's a pygmy people. So yeah, they have a very small little pygmy component according to this amongst the Natufians. But I mean, look at their caucus hunter-gatherer component compared to their pygmy component, their African component. And these are the Scythian components. So look, they even show a little bit of African amongst the Scythians, a little small, little percentage amongst them. I mean, hey, you know, what are the odds on there? Maybe they. I wonder where they got that from, though. It's interesting. If this is accurate and this is true and that's not noise or anything like that, I wonder what component that would be if it's not pygmy component from the Mbute. And if I'm not mistaken, one of them had dark skin. I'm not sure which one it was, but one of the Scythians had dark skin, predicted as having dark skin. I'm not sure if it was this one or not, if it was AS41. I think it was. We'll get to the supplementary material, but I'm pretty sure it was that individual. So but majority of them had intermediate skin, pale, very pale and whatnot. So, but it was just like one individual had like dark skin, and it was actually dark to black, just like a small percentage, like very small. But he had dark skin for sure. So one Scythian definitely was the outlier. And there goes all the mtDNA haplogroups. Non-Scythian being green, middle dawn, late Bronze Age brown, middle dawn bronze, middle Bronze Age being caramel, I guess you would call that, and then the Scythians middle dawn Iron Age being baby blue or light blue. If I'm not mistaken, T1A1, that's definitely interesting mtDNA. I think that's the mtDNA that's found amongst the ancient Israelite sample. At least one of them. All right. I think that I'm right here. Okay. The results suggest that the studied Iron Age groups had multiple distinct Bronze Age populations serving as the main ancestral substantium. Notably, we did not detect continuity between the Bronze Age and Iron Age populations from the same Middle Dawn region. Among all the Scythians we studied, only the early South group shared significantly more alleles with Middle Dawn, Late Bronze Age than with most other tested populations. S era West, since Crimea and Similak likely had ancestors similar to Bronze Age Shurubaniya like groups. In contrast, Bronze Age Andronova ancestry appears to have contributed to the earlier Scythian South and the geographically remote Hyanaboraska group. Our results clearly indicate a different origin and lack of direct gene flow between two components of Iron Age society from the Middle Dawn region. Scythians and settled Similac individuals. Consistent with these differences, previously anthropological studies have highlighted the gracility of the skulls for individuals buried of the Similac, in contrast to the larger size of cranial vaults and characterized by broad foreheads and occiputs observed in individuals from Scythian burial mounds, along with variations in the stable carbon and nitrogen isotope compositions of human bone collagen, as well as strontium isotope ratios, have been documented between these two groups. Moreover, Similac exhibited greater affinity to preceding Bronze Age populations from the Baltic region in southern and western Europe than to the contemporary Scythians of the Scyth Major and all the steppe Bronze Age groups, which were geographically closer. Thus, in addition to the main Bronze Age steppe ancestors, the genetic structure of Scythian and non-Scythian Iron Age tribes can be inferred from the influence of other Bronze Age populations, considering that the Scythians spoke Iranian. Okay, wait a minute. Let me go ahead and read this again. Considering that the Scythians spoke Iranian, it is commonly assumed that they had Iranian or Central Asian ancestry. They just dropped that one in there. Our results show that both the classical period, Scyth Major and S era West, and earlier Scyth South, individuals share more alleles with Bronze Age groups with a high proportion of Neolithic Anatolian ancestries than with the Neolithic Iranian ancestries. A comparison with Iron Age samples are revealed that Scythians presented greater genetic affinity with Iron Age Mediterranean populations than with Iron Age groups from the territory of modern Iran and Turkey. Wow, right there. Bam! Read that again. A comparison with Iron Age samples revealed that Scythians presented greater genetic affinity with Iron Age Mediterranean populations than with Iron Age groups from the territory of modern Iran and Turkey. So these people were migrating and mating with local populations, getting their mix on. However, we demonstrated that the influence of Bronze Age populations with a high proportion of Neolithic Iranian ancestry from the Middle East regions can be traced among the late Scythians, Scyth Crimea, but is absent in the earlier group Scyth South, highlighting the temporal aspect of the genetic heterogeneity of the Scythians. So these people at first were like moving around, not mating, like trying to like stay to themselves as nomads, but some of them mixed with local populations, some of them didn't. Okay. In addition to the main Bronze Age steppe ancestry, Scyth South or Major, Scyth Crimea and S era West also exhibited genetic affinity to preceding Bronze Age Western European, such as Bronze Age England Bell Beaker and England early Iron Age and Baltic Bronze Age populations, but not to Bronze Age Caucasus or Western Asian groups. right there. Bam. But they really don't have any groups of Scythian samples in Western Asia, I want to point out. As far as like the Levant and stuff like that, because they said that they went on trip, like they went on, I forgot how I said it earlier, but they said they went on their adventures, like they went on something. Let me go back to what it said earlier. They went on something. Renowned campaigns in Western Asia and Egypt. So what I'm saying is that they really don't have Scythian samples in the Levant and Egypt. So I mean, until they actually do and say, hey, that these people did come through the Levant and they mingled in, it's like a lot of that stuff is like we don't know. And like I said earlier, Scythiopolis or Bet Sha'an and Israel, yeah, Bet Shean in Israel was known as a Scythian city during the time of the Greeks. So it's interesting. Now get back to where I need to be getting to. Okay. My computer is like running extremely slow now. I need to go up. Up. All right. Okay. The genetic connections of Shumalak individuals to Eastern Baltic Bronze Age groups were also confirmed by outgroup F3 and F4 statistics analyses. Together, these results demonstrate the genetic relationships between Scythians and Bronze Age populations from Western Europe. On the PCA plot, several individuals on the Scyth major cluster, but not of other Scythian groups, overlap with Sarmatians. We tested the hypothesis that some Scythians and Sarmatians may be genetically related groups. The admixture analyses showed a more prominent Neolithic European Anatolian farmer component among Scythians compared to the Sarmatians, as well as other people from the Eastern Scythian world. And most Sarmatian individuals presented more Eastern Siberian and or Eastern Asian or Neolithic Iranian ancestries compared to major Scythians. According to F4 statistic analysis, none of the tested Sarmatian groups were cladded with any of the tested Scythian groups. F3 admixture statistics show no evidence that Sarmatians were an admixture of Scythians and any tested Iron Age groups. The analysis of outgroup F3 statistics revealed that all Scyth major and individuals, including those who overlap with Sarmatians on the PCA plot, share the largest degree of genetic drift with European Iron Age groups and Scythians rather than with Sarmatians. Furthermore, to test a possible gene flow between the individual Scythians and Sarmatians, we use AIM IBD program to search common identity by descent, IBD, chromosomal segments between Scythians and other Iron Age individuals. Although a close relationship was revealed within our Scythian groups, here we go. as well as within previously published groups of the Eastern Scythian world, no long shared IBD segments were detected between Scythians and any tested Iron Age individuals with appropriate genome coverage data. We found only a short common IBD segments of 16 Morgan in link between two Scythians from the Middle Dawn Scythian clan, DS1 and DS9, and two Sarmatian individuals, SGZ001, SGZ002, from the Western Kazakhstan region. Overall, our findings suggest a distinct genetic background for these two nomadic groups. Alternatively, the presence of relationships between some Scythian and Sarmatian individuals may be a sign of admixture between these nomadic tribes. These connections require further investigation with a comprehensive set of high-coverage Sarmatian genomes. Our results reveal multiple origins of first sequence Iron Age groups from the Great Scythia era, mostly from different European steppe Bronze Age populations, and no geographic genetic correlation was found among European Scythian tribes. The earlier hypothesis that the genetic differences in the Scythian Sarmatian world can largely be explained by geography is not supported by our findings. In addition, unlike the Scythio-Siberian tribes in the east, European Scythians do not have ancient Siberian ancestry as their primary genetic substrum. Both ancient Siberian and Eastern Asian components are present in European populations of Scythians at a minor level. Moreover, late Scythians from Crimea do not exhibit these ancestral genetic components. Nonetheless, the ancient Siberian component was found in many proportions in the northern neighbors of the Scythians, specifically in the Hayan-Borovskaya culture population. In addition to the autosomal data analysis, we investigated mtDNA and Y chromosome haplogroups to trace the ancestral, maternal, and paternal lineages of the Scythians and other Iron Age groups. The Scythians displayed notable diversity in their mtDNA lineages, which could reflect social or marriage customs among our Scythian tribes. To identify the potential links between our Iron Age and Bronze Age samples, we compared their complete mtDNA sequences and shared haplotypes. We found 6 matching haplotypes between Iron Age and Bronze Age individuals. However, there were 1 or more mismatches in the complete mtDNA sequences of the individuals bearing these haplotypes. As a result, although these related mtDNA lineages share a deep common origin, there is currently insufficient evidence to support direct maternal inheritance from these Bronze Age to Iron Age individuals. Over half of the mtDNA haplogroups identified among newly sequenced Iron Age samples were distributed across multiple regions of Eurasia during the Bronze Age. However, a high proportion, more than a third, was exclusive to European populations, with an additional 11% found only in groups related to Neolithic Iranian or Anatolian ancestries. These findings align with our findings of our analysis of autosomal markers, indicating a strong correlation between mitochondrial and autosomal genetic markers. Bam. So those saying that sex chromosomes don't matter when it comes to autosomal genetic markers, yes, they do. Mitochondrial and autosomal genetic markers, all of that matters when it comes to population genetics, when it comes to ancient DNA, when it comes to archaeogenetics. So if you're on YouTube claiming otherwise, Shh, you don't know what you're talking about. In addition, a notable diversity in Y chromosome haplogroups was revealed among the studied Scythian groups of different regions and chronological periods. The predominant Y chromosome lineages among Iron Age male individuals in the Middle Dawn samples are represented by two distinct subclades of haplogroup R1A, R-Y2631, and R-Y2. The lineages of haplogroups G2A, G-S9409, J2A1, J-Y26650, and J-FT72594, N1A, N-Z1934, and Q1B, Q-L940 were other paternal lineages present among our tested Scythians, all of them found in different regions of the Great Scythia territory. Because most previously published Y haplogroup data have incomplete haplogroups or haplogroup assignments and low coverage, it is currently challenging to precisely trace the exact origin of Scythians' paternal lineages. However, our results clearly demonstrate a lack of direct paternal genetic continuity between the Bronze Age and Iron Age populations in the Middle Dawn region, as well as between different spatially, temporally Scythian populations. The Y chromosome lineages in the Scythian and settled Similak individuals are not a direct sub-branch of the Y haplogroup found in the Middle Dawn Sarmatian population. Overall, the substantial genetic diversity among Scythians shaped by both major and minor predecessors highlights the existence of complex and currently unstudied contact patterns among numerous ancient migrating groups that contributed to their admixture. The gaps of the late Bronze Age steppe tribes' genetic data now increase the overall ambiguity regarding the origin of diverse Scythian communities sharing a cultural commonality. Whew. Okay. Genetic relation or genetic kingship and social structure of Scythia. Ancient historians describe the unique social structure of certain Scythian tribes, highlighting hierarchical interactions within their society. The royal Scythian tribes were specifically mentioned. And here we looked at genomes of some elite members of Scythian society, the grave goods of Devesta. The burial mound 9 from the Middle Dawn include golden details of ceremonial headdress, such ceremonial headdress, a gold calathas, are attributed only to the richest Scythian mounds. A woman who wore calathas is associated with the Scythian priestesses, indicating Dev 1 was among the highest elite individuals of the Middle Dawn Scythian population. Given the size of the tomb and the numerous artifacts uncovered within the mound, it is evident that one more individual, DS93, was a prominent member of the elite within the Middle Dawn population. The grave goods included horse equipment, gold and silver plaques, and the plates decorated with images of anthropomorphic deities and fantastic animals, and the Scythian animal stock. Paleontological evidence indicates that he exhibited a combination of traits commonly associated with a monastic life in sobriety, including Boresphorus disease, diffuse idiopathic skeletal hyperostosis, type 2 diabetes, and obesity. This historical insight inspired a quest to uncover potential blood ties among the Scythians that may have endured across generations over the territory of Great Scythia. Although we found commonalities in the ancestry of the Scythians from different eras, whole genome analyses did not reveal close genetic kinship between them. However, within the Middle Dawn Scythian population, we detected numerous genetically related individuals exhibiting multiple first and second degree family links using additional mitochondrial haplogroup data, estimates of genetic, sex, and archaeological data. We reconstructed several small pedigrees. These pedigrees compressed one large Scythian clan containing at least 27 individuals of 51 Scythians from Calbanio, I Davistia, V Ternovia, I Gorka, I. Notably, genetic evidence suggests a close relationship between both elite Scythians Dev1 and DS93 from the richest mound. Although over a dozen mitochondrial lineages were equally dispersed into the clan without a dominant maternal lineage, all the men of the family were found to belong to the same Y chromosome lineage of the R1A, 1A, R-Y2631 haplogroup. All right. This finding emphasizes the importance of the single paternal line in the Scythian society, at least in the Middle Dawn region, and expected in the light of similar data obtained from various other patrilineal and pastoralist societies. Most burials, greater than 80% in the Middle Dawn Scythian mounds, were collective burials containing the remains of two or more individuals. Our results did not reveal an association between collective burials and genetically closely related individuals. We observed that offspring were never buried with their parents, but tended to be buried near their parents' burial mounds, often in smaller mounds, although this was not a strict rule. We revealed high levels of consanguinity in Scythian society as estimated from the runs of homozygosity, ROH, among members of the Scythian clan. Over half of their tested individuals exhibited long-range ROH on greater than or equal to 20 cm, with an overall ROH distribution equivalent to that of the parents being second cousins or even closer relatives, i.e., aunt, uncle, nephew, niece, half siblings, and closer. Notably, elite Scythians, Dev1 and DS93, were not the offspring of closely related marriages. Endogamy has been previously documented in some ancient populations. This social practice was common among prehistoric Aegean societies during Bronze Age and has also been described among Carthaginians 700 to 400 Cal BC, Sarmatians, and Scythians. Our findings indicate that at least one Scythian group, Middle Dawn Scythian society, was an endogamous community founded most likely by patrilineal king groups. Analysis of Scythians from other regions reveal no signs of endogamy. Okay. Genetic traces of the Scythians in subsequent populations. the disappearance of Scythians from the historical scene has left unanswered questions regarding the extent of their genetic legacy in subsequent and present-day populations. Some historians propose that Scythians were completely eradicated by subsequent inhabitants of East Eurasian steppes, the Sarmatians. Some researchers have attributed the collapse of Scythia to substantial ecological and climatic changes in the steppe. Our analysis of the medieval populations with F3 outgroup statistics demonstrated that all the Scythian groups share more alleles with groups with ancestors from the Baltic region, Saxon, British Island, and Slavs than with South European and Caucasian populations. Similarly, among present-day Europeans, we observe the highest levels of allele sharing between Scythians and Eastern Baltic, Lithuanian, Estonian, and Northwestern Russian populations. In line with the genomic data, the modern carriers of Scythian mtDNA sequences are mostly found in Europe, predominantly in Poland, Denmark, and the northwestern part of Russia. Well, that refutes the whole concept that Germany was Ashkenaz. I guess Ashkenazi Jews has to call all Europe Ashkenaz. The Scythian paternal lineages marked by Y chromosome haplogroups are predominantly distributed in modern populations inhabiting the southern regions of Russia, the Caucasus, and the Middle East. R-Y2631, R-Y2, R-S9409, G-S9409, J- Y26650 and J-FT72594 haplogroups, and Northern European and Scandinavia, N-Z1934 and Q-L940. Overall genomic analysis revealed that Scythian ancestry is predominantly present in medieval and present-day populations of Europe rather than Asia. Scythian genomes are phenotypes prediction. Herodotus and other ancient authors have described Scythians' physical appearance, traditions, and diet. For example, Scythians were described as blonde or red-haired with gray or yellow eyes and reddish or bronze skin. All right. Figure 5, reconstruction of endogamous pedigrees within the Scythian population from the Middle Dawn Age region. So basically, everything I read, if you want to read this, I don't want to read this right now. I want to get this over with. So, yeah. You can pause the video, read it, and go back over it. That'll work. All right. Greek authors also wrote that wine drinking was widespread among Scythians and mentioned the feminine disease that affected them. Feminine disease. Okay. As pastoral nomads, Scythians probably ate dairy products and meat. We screened the high-quality Scythian genomes for pathogenic genetic variants and variants associated with pigmentation and diet. The effects of genetic variant on genome function and phenotype can vary greatly due to differences in phenotypes as well as factors such as age, sex, and ancestral of individuals. Therefore, we focus on investigating monogenic traits as well as polygenic phenotypes for which predictions with high accuracy could be made. In addition, we developed assays assays using pyrosequencing technology and ultra-short amplicons for targeted phenotype genotyping of ancient DNA. This technique yields direct diploid in genotypes rather than imputed ones and may be applied to test and confirm ancient DNA variants. All right, so this goes into the hair, human eye, hair, and skin color. It says to date, a H-iris plex system for predicting the human eye, hair, and skin color has the strongest prognostic ability, the best accuracy expressed as the area under the receiver operating characteristics curve, AUAC, greater than 0.95 in numerous ethnic groups. Among the various models that have been developed to predict different human phenotypic traits or polygenic disorders, we derive H-iris plex S markers from samples with high quality whole genome data, develop a target assay test in all our samples. All right, I don't want to read all this. Which required hair color. Okay, so eye, predict the eye, hair. Most of the sample individuals were predicted to have brown or blonde hair, with a notable portion of blue-eyed individuals. Several Scythians had MCIR gene variants associated with red hair, freckles, and skin with a tendency towards sunburn. Overall, these results are consistent with the descriptions of the Scythians provided by historical sources. Bam. And I'll go into the supplementary real quick to go into that sample, that one individual that had dark skin. Also, I don't know if I want to cover the whole thing in the supplementary material about the Bronze Age, Iron Age haplogroups, Y in mtDNA, but yeah. So regarding hereditary aspects related to food and alcohol consumption, most Scythians lack both Asian and European variants linked to alcohol intolerance, ADHIB and ALDH2 genes, and lactose persistence, MCMS, LCT, Loki, respectively. Given their genetic data, they most likely did not experience any negative effects after consuming alcohol. Uncertainty remains surrounding the culture of milk consumption among Scythians, the genetic factors underlying these practices, and the mechanisms of lactose persistence in Scythians. Moreover, milk may have been fermented for consumption as alcohol. Nevertheless, our results suggest that Scythians did not acquire a notable portion of lactose-persistent genetic variants from European farmers, in whom these variants are under stronger selection. In the present study, pathogenic genetic variant screening of the Scythians' genome did not reveal the definitive genetic cause for monogenic obesity, diabetes, hormone imbalances, or sexual dysfunction. However, it is important to note that common forms of these traits exhibit complex etiologies, making the identification of their genetic factors particularly challenging. According to our criteria, we detected 37 pathogenic or likely pathogenic variants among the tested IA individuals, Scythians from the Middle, Don, and Premier regions, as well as individuals from the Similuk and Kabon population harbored the P.His 63 variant in the HFE gene. This variant is linked to clinical hemochromosis with reduced penetrance and conditions affecting iron metabolism in present-day humans. Of the present-day individuals, homozygous, and for the okay, I don't want to read all of this. That's the mutation contribution. So their diet mutation contribution could contribute to the Scythians' ruddy or bronze skin, reported by ancient writers. Okay. So their meat-heavy diet, meat-rich diet. Okay. Let me see. I don't want to read that. This goes into the low sugar diet consumed by the individual's homogeneous. Okay. That goes into the fructose intolerance that it's mentioned and found in some Europeans. This is what it's talking about. They do populations to see of the fructose intolerance, mutation dietary preferences among middle dawn Scythians, and also this includes modern day populations. Yeah, I think I'm done with that. I want to go into this. So this is the supplementary materials, genetic history of the Scythia. So this is the part that I'm talking about. They mention Assyrian writings, right? But they don't mention exactly what the Assyrians referred them to as. So yes, the Scythians and the Assyrians had contact, and the Assyrians had their own word. They didn't call them Scythians. Scythians were something that the Greeks called them, but they didn't. But the Semitic speaking people did not call them that. They called them Ashkenazi, something like that. They didn't call them Scythians or Scythians or whatever the ancient Greeks called. And it shows that the civilizations of ancient Greece, the Middle East, and China were substantially influenced by steppe nomads. This is why the peoples of the northern Eurasia first received a historical voice in ancient written sources. The historical Scythians, known from ancient Persian, Greek, and Assyrian writings, are associated only with the western part of the Scythia Siberian world, the cultural and historical region of the northern Black Sea coast, and partially the northern Caucasus. The definitions of the term Scythians and Scythian archaeological culture remain complex and debated. Here we understand that Scythians as conglomerates of tribes that left numerous burials, predominantly in the form of mounds, featuring inhumation rites across the steppes and forest steppes territories from the lower reaches of the Danube to the Don, as well as in the North Caucasus. These burial grounds are mainly associated with the paramilitary part of Scythian society. I'm going to go to the haplogroups real quick. And this supplementary material is so big. There's so much material that they cover. There we go. I had it, but I didn't have it. Okay. All right. Let me see. So they had a total of 131 dually sequenced Bronze Age and Iron Age samples were used for mtDNA analysis. They performed analysis of a complete mtDNA sequence, mitochondrial haplotypes, and mitochondrial haplogroups. And I'm really want to get to this part. It says 3 of them that belong to haplogroups V7A, HV, and H1B2 were found among modern Western and Eastern Slavs, suggesting that the Scythians could share maternal ancestry with present-day Eastern European populations. Another completely matched mitochondrial sequence related to the H13A2 haplogroup was found in one individual from present-day Greece. Says, interesting, Scythians complete mtDNA sequence of mitochondrial haplogroup U4B was found in many individuals of present-day Zoroastrian Parsi population from South Asia, India, and Pakistan, and thus is population specific. However, given the current data, it is impossible to determine whether modern Parsis descended from the Scythians or whether they shared an earlier common ancestry. Are the Y chromosome haplogroup analysis. This part is very, very interesting that I want to bring out specifically for the Jews that carry R1A. That specifically is the Ashkenazi Levites. Even Netanyahu, Benjamin Bibi Netanyahu, Malakowski, his original paternal last name, he carries R1A. His son Benjamin, I forget his son's name. If I can pull it up for y'all, but his son tested on family tree DNA. I think last year or 2023. Yeah, it was 2023 if I'm mistaken. Let me pull it up. Yep, it was December 2023, around that time frame. Let me see. All right. So he belongs to R-M198. That's just the haplogroup that they assigned him. I'm assuming he took probably a basic Y111 or the basic one. I think it's Y12 or something like that. Or yeah, I think it's Y12. But I think that's the basic one he took. As you can see, I made my comments over here to the side. Because this person said he wasn't Ashkenazi. I'm like, Ashkenazi for sure. You know, the Semitic word Ashkenaz were actually referred to the Scythians. The Scythians carried haplogroup R1A. The same with the Cimmerians, which were called Gomer by Semitic speakers. This means that Netanyahu is a male descendant of the non-historical Japheth. Yeah, so actually reference, yep. So there you go. And to show the migrations for y'all, for that don't really know R1A like that, specifically leading up to the branch that the Ashkenazi Levites carry. I'm going to go ahead and play this on the Globe Tracker Discovery. So let me go ahead and play this. So we have R1A already there. Let's continue this. Let me go ahead and play this for y'all. So it's interesting. If you notice that, it's a huge time gap that they took from R-M19 up here. And I think is that Poland, if I'm not mistaken? I think that's Poland. Ukraine. So Ukraine is where R-M19 8 mutated in. That's the SNP. That's where it mutated in. So I understand that's a long time ago. I think that was 6500 BCE. But we'll continue on to see exactly how this gets all the way over here, going into Eastern Europe in the Balkans. I think that's Hungary right there. It's going to jump all the way back to Ukraine area and into Central Asia. Watch this, y'all. Y'all see that? Y'all see that huge time gap right there? They went all the way from Ukraine all the way back into Afghanistan, yo. Crazy. I want y'all to like check this out for yourselves, or make a comment what y'all think about that. Where is how is that a huge time gap of migration? So you telling me these people went all the way from Ukraine? It took them forever to do this migration right here, but they went all the way from Ukraine to this paternal lineage, going all the way back to Afghanistan. That's crazy. Like, look at this shit. All the way to Afghanistan. Now check this out. So R-Z93 is the SNP that most of the Scythians were downstream, underneath. They were underneath Z93. Specifically Z2122, which I'm going to show back again on the supplementary material. This is important. Very, 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 very important when you're trying to understand these different paternal lineages and whatnot. Let me go ahead and get this for y'all. It's going to be under the Iron Age. Right here, it says the Scythian associated Y chromosome haplogroup R-Y2631 lineage belongs to the ancestral R-Z2122 clade, which is a sister clade of R-Z2125. Now go back to the Google Chrome. Okay, so check this out. You see this red? This marker right here is going back into Europe, right? And when it's doing that, it's going to avoid Israel and area. It's going to go above, like in the northern part of the Levant, but it's going to miss Israel. But this blue one right here, this R-F1345, that is the mutation. That's the SNP under what the Ashkenazi Levites carried. This is the father of R-Y2619, if I'm not mistaken. I believe that's correct. 2619. 2619. Yep, correct. That's it. Watch this, y'all. Look at the time periods of when it goes into the land. So right here, this is 1300 BCE. 1300 BCE, right? Check this out. It's already 700 BCE. They're still not in Israel. Israel's down here. They're still not in the land. Still not in the land. But here we go. All these Scythian descendants going up into freaking through Iran, through Iraq, through up into Turkey, the Caucasus and whatnot. Not over here. And so if they had a migration into Egypt at one point, I don't know when it was, because this is not showing this. because this blue line should be coming all the way over here. That is the mutation that that produced the lineage, and I'll show it for the Ashkenazi Levites. There it goes. Look at the time period, y'all. 600 CE. It's finally making its way into Palestine territory, Palestinian territory. Finally, it's arriving. 800 CE, y'all. 900 CE. Very important when you're looking at these mutations of where these paternal lineages come from and whatnot. It spread all throughout Europe before it ever landed into Palestine, Israel territory. That's some truth for y'all. Bam. Oh, we got him. I got him. It's a wrap with this. So they never came into the land at no 1000 BCE to be no ancient Israelites. Look at that. They were never there in the time of the ancient Israelites. And like I said, it's this blue line right here. That is the this is the paternal lineage that produces the R-Y2619 that's found amongst Ashkenazi Levites. Yep. All right. And so this is the Scythian one. This is the R-Y2631. This is the migration route from 1500 BCE. Let's go ahead and see his lineage. Man, the number of the people in the video went up to 10 or something like that when I brought up Ashkenazi Jews, man. I saw that. It peaked up there. All right, so here it goes again. Going from Afghanistan, spreading westward. So they're saying by 1200 BCE, it was already in Eastern Europe, already through the Balkans. There it goes, y'all. That's the Scythian lineage right there. And R-Y2, I could not use Globe Tracker for that. So it's going through the Caucasus. It went from Romania to the Caucasus, through Ukraine to the Caucasus. Check this out. So we're at 700 BCE. Spread around the Black Sea area, it's really just like the paper stating. It's pretty spot on. And that's it for that one. More data, more populations of ancient Scythian SNPs, complete genomes where they can get the Y-DNA and whatnot. I think this will update this as far as where they can continue to spread into. But yeah, that's the paternal lineage though, what I was talking about. Oh, let me go ahead and show that real quick. I want to show that real quick. Give me a second. I'm going to go ahead and turn this presentation, this one off. Let me get into this real quick. See if I can pull it up. Yep. Okay. So I think it's Y2631. 6, 1. All right. Check this out real quick. So the rabbinical, so these are both Jewish. This is the Ashkenazi Levite. This is rabbinical. Okay, we're gonna go ahead and get into this. Watch this. This is the R-Y2631 that was found for this Scythians, right? Check that out. They share R-Z2122 as a common ancestor. Can't make it up. So between them coming from Afghanistan and spreading westward and missing Israel and not until freaking the Common Era, you can't make this up. These people have really nothing to do with Israel when it comes to the Jews, and they do share a common paternal ancestry with the Scythians. It's no lie. I was spot on in my original 3 videos that I did this for sure. And more ancient DNA will prove what I said about the Scythians and the Ashkenazi Levites or the Ashkenazi Jews carrying R18. And they're not the only ones that carry too. I found that other Jews, I think they're from Iraq and India, carry R1A too. So Mumbai Jews. So there's other Jews besides Ashkenazi Jews that carry the similar Y DNA that the Scythians carry. All right. There it is. R-Y2631. They share a common ancestor. Okay, shared ancestry. Let me see something real quick. Go ahead and block that. I want to get into the other one. There we go. There we go. The Howitz. Yep. All right. So it says the predominant Y chromosome haplogroup among Iron Age men from the Middle Dawn region is represented by 2 distinct subbranches of the Y haplogroup R1A. The first and most prevalent subbranch is, I'm not saying the whole thing, is R-Y2631, while the second subbranch, you can read it for yourself, R-Y2. Both are derived lineages from the R-Z93, whose subbranches predominantly occur in present-day Central and South Asian groups, in contrast to its sister branch, R-Z282, which now common in Central and Eastern Europe, with the subbranch R-Z280 dominating among Eastern Slavs. Importantly, neither of the 2 Scythians branches was a subbranch of the haplogroup R-Y934 that was present in the population of the Don Forest Steppe Sarumnaya culture, Middle Dawn, Late Bronze Age, in the same region during the Late Bronze Age. It is important that the R1A's haplogroup was not observed in any of R sampled ancient DNA Central and South Asian until after 2000 BCE, and it arrived to this region later with migrants from the Eurasian steppe. So exactly the little map I showed y'all with the globe tracker, that's exactly what he's talking about. It went all the way from Ukraine, all the way up there into freaking South Asia into Afghanistan, or Central Asia, South Asia. The Scythian associated Y chromosome haplogroup R-Y2631 lineage belongs to the ancestral R-Z2122 clade, which is the sister clade of R-Z2125. Notably, the R-Z2125 clade is ancestral to the R-Y934 lineage observed in the Late Bronze Age group. Both sister clades R-Z2122 and R-Z2125 belong to the Y chromosome branch of a branch R-Z2124. Yep. And you see how, for those that don't know this stuff on why DNA is important, what did they use, y'all? Y full and family tree DNA. What did I use for the neurorat sample in my previous video? Y full and family tree DNA. Some people just don't know how to do all this stuff. It's just way past their understanding. It's really funny. But let me go ahead and pull something up real quick. I want to pull up the Y tree real fast. And before I do that, here, check this out. So if y'all don't want to watch my previous videos, this is the place that I'm talking about. Beit Shean, Scythiopolis. There you go. During the Hellenistic period, the settlement was known as Scythiopolis. After the region came under Roman rule, Scythiopolis gained imperial free status and was the leading city of the Diapolis. So the thing is with this is that there is no ancient DNA from Scythiopolis. I guarantee if they find some ancient DNA, some genomes, Y DNA, specifically amongst these men that were there, I guarantee they'll be R1A2. 100%. I don't think the Greeks called them Scythiopolis for no reason. The Scythians. And like I said, in the book of Colossians chapter 3, verse 11, it clearly talks about them as being Scythians. And also, I think in the book of Judges, in the Septuagint, yes, in the Septuagint, you can also, oh, my bad, here we go. My bad. So here we go. This is what I was talking about. Bam. There goes your sources right there. But in the book of Judges, chapter 1, verse 27, look what it talks about, which is a city of Scythians. So Bethshan, right here, city of Scythians, bro. Look at this. And Jews, when I was on Clubhouse one time, it was Ashkenazi Jews. They try to say, oh no, that those were military age men that retired and just settled there. Yep. Okay, if you say so. But we know for a fact that those were Scythians, those were foreigner Scythians that settled in the land. But when you come on here, they won't show that as far as them settling in the land. But it makes this look bad because they're saying basically that they don't belong to, they were never in the land during that time. When I showed Globe Tracker, it didn't show them ever coming into the land until the Common Era. So that's a huge blow on their part. And that's about it. I hope y'all gained a lot of information from this video. As far as the Scythians, like I said, I've covered them before. Please do to make sure to go watch the original to have a full understanding of who the Scythians were. And what is the connection between Scythians and the Ashkenazi Jews, Ashkenazi Jews? For the most part, no, Ashkenazi Jews don't have ancestral components like that to show that they're descendants of Ashkenazi, of the Scythians. However, according to this, which is something interesting, maybe they do. You know, maybe when they did the PCA, let me pull this up again. before I end this video. Right here. I mean, maybe there's something more to look at. And then I'll pull up I want to pull up the Y tree real quick for this lineage so everybody can see what I'm talking about. All right, so this is the Y tree. Make sure this is situated for y'all to see it. All right, so this is the Y tree, right? And we see the Ashkenazi Jews carry this, and this is a downside plate of R-M198. Where's the other one at? Let me see something real fast. Here, let me close this up. I have to sign back into family tree DNA real fast. I can't stand that they kick you out like as soon as possible. Like they make sure nobody steals your stuff, which is great. I mean, that's good. But it's kind of annoying that it'll kick you out fast if you're not active on it. All right. Yo, this is the most annoying thing ever. Why isn't it working? Give me a second now. I'm pulling it up real fast. It all messed up for me. All right. All right. All right. Why is it messing up? Give me a second. Give me a second. Ay. They don't want me to show this at all. Like it keeps on asking me to sign in. Like how many times do I gotta sign in? Like, damn, I done signed in 5 times. Wow. Crazy. Oh my goodness. All right, finally. Nuts. Absolutely nuts. Anyways, y'all, this is it right here. This is the R-Y2619. The mutation occurred 700 CE up in, it looks like Ukraine area. Yeah, that's Ukraine. So northwest Ukraine. Right. So let's go ahead and go for the mutation ride. So it came down from R-CTS6. So this is what I was talking about earlier, how they split off coming from Afghanistan, right? And then they went through Iran. They went through Iran. Let me go ahead and play it real quick. Let me so y'all can see this. Y'all can see what I'm talking about. I ain't making this up. Watch this real quick. There we go. You see how they went all the way from Ukraine into Afghanistan, going through Iran, clearly missing Israel. 1000 BCE, they were in Syria, through Anatolia, going in through the Balkans, or to Eastern Europe. Look at that. Bam. It never was in the land of Israel, according to this. Let's see this green mutation, when that occurred. Let's see that, when that occurred. So this is R-Y2630. I'm going to look into this one because it's showing that's 1600 CE. So around 1900, look at that, 1900, it came into the land. 1900 CE, it came into the land. Now I want to see something real quick. Let me see if they have this snip on here. They do. Okay, so they do, and you can see that it's Y2630 is underneath Y2619, right? Perfect. You see it, Israel, Ashkenazi Jew. Y2619, Germany, Ashkenazi Jew. Same Ashkenazi Jew from Israel. CTS, which I just showed. Ashkenazi Jew under Y2619 is found in Turkey. Keep going backwards. F1345. Found amongst the Avars. Mongolia, Hungary. I think this is a Avar sample too, if I'm not mistaken. I'm curious. But no, is that modern? Ancient. I don't know what this is though. I don't know if that's an Avar or not. Kazakhstan, Kyrgyzstan, China, Kyrgyzstan. So this is part of the Eurasian steppes. Okay. Go back. S4576. And I'm about to end the video. So if you stood this long, I appreciate it. If you enjoyed me reading the material and giving you all the insights, please like, share, and subscribe. And if you feel like you have any beneficial feedback to leave me, please do so in the comment section. I much appreciate it. Avar. Okay. Let me see something, because there is Z1, 2. All right. And see, this branch right here was mentioned in the paper, in the supplementary material. Go ahead. So Z2124 was widely distributed among Eurasian steppe during Bronze Age and was found among numerous populations of, they misspelled that, Androno do. I think they misspelled. If they didn't, Adnorno, I don't know how to say it, and Shurumna. Yeah. Cultures from Russia and Kazakhstan territories. Its derived clade R-Z2122, which was ancestral to Scythian R-Y2631, were found in Bronze Age samples as Simya terminal culture, Yagni, Russia, in the steppe groups of Kazakhstan. Yes, sir. And look at there. Bam. Just like it's saying. There she goes. Ba 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 ba. Yow. And see, like I mentioned, it's not just Ashkenazi Jews. There's also an Iraqi Jew. And I think there's like there it goes, a Mumbai Jew. And it looks like it's found in Israel. Both of these samples are found in Israel. ISR01, Israel 42. So I think both of these individuals live in Israel. And just like I said, Afghanistan with mutation. Look at all these Afghanistan components. All right, check this out. Were the Scythians in Afghanistan? Bam! So it's interesting that their lineage migrated from Central Asia into the region of modern-day Afghanistan, Pakistan, and northern India beginning in the mid-2nd century BCE. These migrations persisted for several generations until around the 4th century BCE. But why did it show that that mutation occurred in Africa? How did they migrate there? So they back migrated into the land? Is that what I'm getting? Is that what they did? They back migrated into the land? Because according to this, they went to Afghanistan a long time ago. Look at that. So that's Afghanistan right there. R-Z93 is in Afghanistan. That's Afghanistan. That's actually where I was at in Afghanistan. I was deployed in that area. I was right here in Host Province, right there. So I was right here. I was deployed right here. And I think I went through this area too, if I'm not mistaken. I don't remember. But yeah, I know I was all in the Host Province. But yeah, look at the migration from Afghanistan going outwards. So we see it go through Afghanistan, go through Iran, go through Iraq, and spread out through Syria into Anatolia. Now let me see something real quick. So that was 1200 BCE. Like I said, they're showing it coming in late 1900, 1700. There you go, 1700 CE. but it was already through Europe. I want to see this prayer real quick. It's like watching the forecasting for hurricanes. Because I'm not expecting to get a hurricane soon because of the Saharan dust storm that's coming this way to Florida. Hey, look at here. It's like watching a forecast of migration. Bam. All right, y'all. I think that's a wrap. You know, it's been great. It's been great, and I might do one more video before the semester starts, unless there's a paper that's released. I think my last video that I'll do before the semester start will be the Thracians. And until a big paper comes out, I'll be busy with the semester and whatnot. So I'm gonna leave it there. Hope you enjoyed. This has been Biblical DNA, and I'm out. Peace.